we're heading to University of Miami to meet with our team at the Sylvester Cancer Center, and I'm going to take you guys along with me. Hi.
Dr. Ashish Shah. I'm one of the PI of the section of virology and immunotherapy. We're working on trying to figure out how viruses interact with brain tumors and different ways we can target them with different antiviral therapies and other ways we can harness the immune system to try to improve the immune response against brain tumors. So this is our lab. Outside of our area, this is where all the work is being done. So this is Jaleesa. Jaleesa's a star medical student, Miami born and raised, and she's working on what are you working on right now? So she's harvesting some GBM cells, and we're trying to see how some immune cells interact with the GBM cells in culture, and these are some of the proteins we're looking at in culture, and we can visualize them with live cell microscopy here.
Okay, great. So hi, my name is Michael Ivan. I'm the PI of the Brain Tumor Invasion Lab and director of our skull-based program. And this is Tanya, who's head of our lab here, and we'll be showing you around.
This is our tissue culture room where we're growing the cells, which derive from the actual patient's tumors, and they're growing right there. That we imitate the brain environment and try to study the invasion of the cells. So, which is obviously it's hard to do in a lab in a dish, but we try. And we are investigating different kind of pathways which force the cells. Well, actually, not force, but kind of imitate the invasion and asking questions: why do they invade? What can they? What molecules? What methods to stop that invasion from happening? So.
Talked this morning about understanding where the tumor is and how it invades and kind of destroys some of the brain functions and the networks. And the other half of the spectrum is how to protect those networks and how to preserve the function of a patient. And that goes well beyond just preserving their movement of their arm and leg or preserving their ability to talk. So we're now trying to go into the realm of understanding how somebody thinks and how their anxiety is and their depression and their ability to do higher order cognition and leadership skills. How do you preserve that during surgery? And we are looking at the connections of the brain to understand that. And so we're working with two different companies. One is called Omniscient, that is able to use advanced MRIs such as resting state fMRI and tractography to kind of predict where these large areas of the brain that are connected are. And they're complex because now it's not just one tract going from A to B, it's like 50 areas connected to another 50 areas, and in everybody it's different. And so we use AI to map that out and to visualize it, but then it's tricky to see in 2D. So now we've turned to kind of taking that plan and then using it with Metavis, which is a 3D augmented reality company, to actually plan it out, see it in 3D, and then we could take this kind of final plan, bring it into the operating room, and really understand how we're going to approach the tumor to minimize any risk to those areas that are really significant to a patient.
Just to add to that, we're looking at a holographic scan. This scan can be registered to the patient anatomy. So there's a couple ways to do that. Now we have automatic facial registration if the patient's lying up to do that with. In addition to that, we could do the trace registration or landmark. So there's multiple ways to do it. And then once you have the hologram registered as a patient, it follows that patient. So there's a reference tracker that basically is keeping tabs on where that volume is anchored. So if the patient has to move or anything, it is the scan will be anchored to the patient as well. What you're looking at is a burn-in image. So we actually pull this from PACS, so we're able to get the information directly off of the hospital's information system, load it on, and get registration done within the minutes for the patient, depending on what the case is. So,
yeah, let's look at it in 2D first, maybe. This is post-op. This is a patient here with a glioblastoma recurrence. and we were trying to push the limits and kind of further the resection here. We could see there's some recurrence superiorly and anteriorly, but we know that there's certain areas that are critically important. And so here we're seeing in blue where the sensory and motor tracts are, and if we, this is the combined one. And now we can start adding some of these other networks like the dorsal attention network and the visual fibers and the visual tracts, and we can start seeing where are the critical areas around the resection cavity that are critical to preserve. Some of them we could map out by stimulating during surgery, like the motor area, but some of these other ones, like the dorsal attention network and visual fibers, are much more challenging. There's no way for us to check that or double check it during surgery. So we're just going based off of the anatomy, and this really helps us understand what areas we could be aggressive with. Like, for instance, up top here and medially, there's no reason that we need to leave any of this tumor behind. But if we look anteriorly, there's almost some overlap here of where some of the sensory motor fibers are, and we have to be really careful where that is. Inferiorly, if we go down, there's more visual fibers that are coming around the outside of the capsule, and that would be critical to understand.
Back here, you want to tighten that, and then you have it sit in front. Tilt the visor down towards you.
I mean, tilt it down. I think it's on. No?
Tilt the visor. There you go. Towards it's on, right? So now what I'm going to do is I'm going to connect the headset. You're going to see.
This is cool. I'm supposed to sit here?
So yeah, anywhere, because you're going to basically place that volume in your 3D space. So you're going to
Whoa. Wait, I was not expecting that.
Step back. It looks like you do have it anchored already.
Then you can grab it and snap it with your pincher fingers.
Oh my god, whoa.
The tap to place, you just push right through it. So you poke all the way through. There you go. So when you reach your hand out, you'll notice there's a cube. And you can manipulate the volume, move it. You can introduce your second hand and stretch it out. Just get familiarized with the gestures, and when you're done moving it, just release your hands and drop them to your side. You can walk around the volume.
A little further away.
Are those the eyes? Whoa.
And when you're done moving it, you just release. It has to be, there you go.
Tilt it. I guess it's the bottom. Whoa. There we go.
Stretch it out. You can enlarge the hologram. You can shrink it. Do it. So once you have it anchored and then placed somewhere.
So my name's Britton King. I'm the Executive Director of Development at Sylvester Comprehensive Cancer Center. And I want to talk just for a minute about the importance of private philanthropy. A lot of our researchers, physicians, scientists are brilliant, and it takes them 8, 9, 10 months to write a research grant. Then they wait another year to find out if that grant's accepted, and only about 7% of those grants are accepted. With private philanthropy, we can speed up research leading to patient care, research leading to patient cures. So private philanthropy really speeds up the science and makes a huge impact.